Hello everyone, today we're doing the nitrate reduction test. This test is done on gram-negative organisms that use nitrate as the final electron receptor instead of atmospheric oxygen in the cellular respiration process. What we're looking for is basically, this is a nitrate broth tube. Okay, and this has been uninoculated thus far. It has nitrate in the broth. And the Durham tube here is upside down to see if there is gas production by the organism after incubation. So when you're looking at inoculating your tube initially, you want to make sure that it looks clear and that there is no gas here. If there is gas here, that might be that you have growth in here that you don't realize. Okay, so this is showing that this is a good one to inoculate. So what we're going to do first is we're going to inoculate some tubes. We have two tubes here, one for Moraxella catarrhalis, the other for Neisseria meningitidis. You can use the nitrate reduction test on more than just the gram-negative cocci. We have gram-negative rods as well that we use in class, but currently we're in the first grouping of organisms, which includes Neisseria and Moraxella catarrhalis. So the problem with this whole shebang is that Neisseria also looks like Moraxella, and they also inhabit the same locations in your body naturally. So we're looking at the mucosal membranes, especially the upper respiratory tract, the mucous membranes of your upper respiratory. So if you look at your decision tree that we went over in another video, there was accidentally a typo down here of Moraxella and Neisseria. Neisseria should be nitrate reduction negative, and Moraxella catarrhalis should be nitrate reduction positive. I accidentally made a mistake when I did that the first time. So the Neisseria we have in our lab is Neisseria meningitidis and Neisseria lactamica, and the Moraxella we have that is the only Moraxella species that is gram-negative cocci, diplococci, is the Moraxella catarrhalis. All the rest of them are cocobacilli or rods, and they're not really very virulent, and you don't see many infections from them. So usually if you see other species of Neisseria other than lactamica and meningitidis and gonorrhea, or other species of Moraxella other than catarrhalis, then you're expecting that that would be a contaminant. So although this grouping of organisms that we have at the beginning of the lab class does not include the gram-negative rods, like the Enterobacteriaceae and the non-fermenters and the rest of Moraxella, we're just going to focus on this, and this process can be applied to either one of those groupings, okay? It's just the test itself. So the way that you can inoculate an organism into the nitrate reduction broth can either be from hold on, I'm getting it, either growth broth. This is thio, and we have an organism that was in there from a stock culture. Or you can take 2 drops of that, or you can take a loop and get a couple colonies or a loop full of the organism and put it in there. So that's how we went. We're going to go ahead and do Moraxella. So this is a stock culture. Notice I've been using it for a lot of stuff. That's why there's a lot of it missing. So we're going to take a loop full of this. I don't know why it's not focusing. Sorry about that. There we go. A loop full of this, okay? And we're going to inoculate it into the Moraxella tube. Okay. This is going to end up being loosely capped and put in a non-CO2 incubator for 24 to 48 hours. Some organisms may actually require up to a week in incubation in order to get the results that are expected in the nitrate reduction test. So but we're just going to do it for 48. Okay, so see that I tightened it all the way, and then I did it loosely back. Okay, about a half a turn. So now we're going to do the next one. So this one's going to be Neisseria meningitidis, and we're going to do it from a thiotube this time, so you can see that. Lucky you. You want to go down where there's a lot of growth. Luckily, there's a lot of growth in here anyway, so that's all right. And this is going to be capped and put back. Hold on. Okay, so I have my pipette full here, and I'm going to put it in. We're going to do 2 drops of the, this is not focusing very well at all. 2 drops. There we go. This one had an air bubble, so 1, 2, we'll do a half of 1. Make sure we get all that. Okay, so again, I don't know why you're not focusing. There we go. We're going to tighten it all the way and then loosely undo the cap. All right, these are going to go again in the non-CO2 incubator for 24 to 48 hours. This one is your control tube. This is uninoculated. It's going to be incubated and tested with the reagents just the way that you would test the test organisms to make sure that the process is going correctly, including that there is no contamination of this tube itself. Okay, so non-CO2 incubator for 24 or 48 hours, and I'll get back to you. Okay, so we're back, and we did our 24 to 48 hour incubation period. We did 48 hours. You can notice that there is growth in the tube of each of them because they're cloudy as opposed to the uninoculated one, which remains clear. There is a little gas production in the Moraxella tube here, okay? So if you were doing this on non-fermenters, the GNRs, you would end up, if you see gas there, that was done by some other type of process than necessarily the nitrate reduction process done by Enterobacteriaceae. Okay. So, which are fermenters, or that's in another video. Okay, so they do ferment sugars. It all depends on what organism you're talking about as to what sugars it does ferment. But anyway, so that's another test. So these are the 3 that we did. What we're going to do now is hopefully keep everything in focus because I'm having trouble with that. This is reagent A. We have 3 reagents we're possibly going to use. We have reagent A, reagent B, those are liquid, and then we have zinc dust, which we use only if necessary. So let's talk about this. In reagent A, we have sulfonilic acid. I'm so sorry. Sulfonilic acid, and if nitrite is present, because nitrate was reduced to nitrite, the nitrate was provided by the broth, and if the organism reduced it into nitrite, which is NO2, so nitrate is NO3, nitrite is NO2. So if the nitrate in the original broth was reduced to nitrite by the organism, then reagent A here, the sulfonilic acid, is going to end up forming a diazonium salt with the nitrite that was produced from the nitrate. Okay, I know this sounds very similar, all of it does, so I'm trying to say it slowly. So then if that's the case, when we add reagent B, and you cannot do it out of order, it has to be A, then B, reagent B has alpha-naphthylamine in it, okay? That's what this actually is. And when you end up putting this in with that diazonium salt that was made from the reagent and the nitrite, if it was present, you end up making a red water-soluble azo dye. Okay, so if the nitrate was reduced to nitrite and both of these reagents were added. If that was the case, it turns red. If there was no color change after you added these first two, then that could mean two things. It could either mean that you still have nitrate in the broth after, the growth of the organism, and it was in place with the nitrate in the broth. If it reduced it all the way past nitrite, then you might get ammonia, which is NH3, nitric acid, which is NO, or dioxyhydrazine, N2O2. Okay, so those are all products past nitrite reduction. Okay, so that could be why it's still the same color and didn't turn pink. If it turned pink, that meant that nitrite was present, okay, and the nitrate was reduced to nitrite. If there was no pink present, then that means it either went past nitrite to those three compounds that I told you about, or it still has nitrate there and it's a negative, okay? So to figure out which one you have to add the zinc dust only if there was no color produced, okay? So we want to find out, is it a true negative or is it actually a positive? It's just past the point that we expected it to go, okay? So if we add the zinc dust here and it turns red after the addition of the zinc dust, then it's a true negative, and that means the nitrate is still present in the broth, okay? So we are hoping that these are going to go right, and the Moraxella should be positive, the Neisseria should be negative, and the obviously the uninoculated tube should be negative too. And still no gas production here in the Durham tube, the upside down tube. Come on and focus for me. Okay, so that's good. That's showing that that's still good. All right, so we're going to get started. You're going to use a normal transfer pipette for this whole process with the two reagents here. Okay, so we're going to uncap everything except for the reagent. The reagent is flammable. You want to keep it in the flammability cabinet. My mistake, sorry, it's not flammable, it's corrosive, but you don't want to breathe the mist and the vapors, okay? So you want to keep your reagents capped, but you can have your specimens open, okay? So hold on just a second. Okay, so when we add these, we want to set our timer for 3 minutes, because the color change you're waiting for should happen 1 to 3 minutes. All right, so we'll end up using our timer. The other time we have set is for the zinc dust, and that's setting 2, and that's for 10 minutes. But we're going to do this one first, okay? So let's get this where we can possibly see it. All right, so I have all of them uncapped. You can use the same pipette as long as you don't touch the mouth of any of these tubes, okay? And you don't take it down into the actual tube. So we're going to swirl. Remember, this is a health hazard, so you want to keep it uncapped as little as possible. So I'm going to pull some up. We're going to do 5 drops in all of them, okay? It can also be 0.5 mils, but I'm going to do the drops. So let's see if we can, there we go. All right, so 1, 2, 3, 4, 5. I don't think you can still see this. There we go. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Whoops. Okay, so the excess of the reagent that I still have in my pipette, since it's still clean, I can put back in there. All right. Close it up again. So we do that one first. Then we're going to add reagent B. Same thing. You don't want to leave it open. because of the health hazard. So you're going to end up swirling it, uncapping it, same thing, 5 drops. Gonna pull out a bit. So this one looks different. It's like a normal broth color, whereas the other one was like a reddish brown. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 2, 3, 4, 5. All right. Put the rest back in. And we're gonna set the timer to start going and look for a color change. I'm going to recap while we're sitting here. Okay, so let's look so far. So far, do you see any difference? On the camera, it looks like this one's turning pinkish. In real life, I can't really tell. Oh, there it goes. It looks like it's starting. All right, I'm gonna let them sit. And, whoops, no. As it's going, I'll do that off camera and then come back. Okay, this is after 2 minutes so far. We can see this turning pink visibly, and I'll keep letting it go for the rest of the minute and I'll come back. All right, so we're in the final countdown. Pink to red is positive, and no color change would mean that it's negative. So we do have pink in the Moraxella catarrhalis, which was expected because that's supposed to be positive. There was negative so far here. Okay, so again, we've talked about previously, earlier in the video, that if there is no color change, we don't know if that's a true negative or not. Okay, so the next thing we will do is add our zinc dust. Okay, and it does look like metallic dust. Okay, so don't leave it open or inhale it or breathe it in or anything like that. You'll end up getting it up your nose. So what we're going to do is we're going to use this little clean spatula guy. You can also use a stick. This is the stick that you would use. It's a clot detection stick. You know, you put it in and you check for a clot in a whole blood tube. So I'm going to try to show you both types of processes. I'm going to uncap these really quickly. We don't need to uncap the Moraxella because we know that's already positive. So we're just going to do the 2 negatives. So I'll try to do the one with a stick and then one with the spatula. Let's do the stick one first. So the idea of the stick is that you can literally put it into the zinc. Okay, you just stick it in there, and whatever comes out on the stick should be enough to make it change color appropriately. And anyone who uses this type of technique likes the idea that you can leave the stick in there to show that the process was done. However, does not look like I really got much on there. It's changing a little bit. I'm not sure you can see that on the video. There's, see the little bit of pink? So that's showing that it was a true negative, right? Because if there's a pink color after addition of the zinc dust, then that means it was a true negative, okay? There is no reduction of nitrate past nitrite. So really, that's all you need is that little bit that was on the stick. I'll show you the spatula version. This is a little more challenging because, you know, everybody wants to dig a lot out of there. Okay, so trying to focus here. There's just a little bit in there. Come on, camera. All right, well, I don't think that you can see that very well, but there's only a little bit in my spatula. Okay, it's supposed to just be a pinch. I shoved it in there. Now, I definitely need to clean this from the reagent, but also I just touched the mouth of the tube, so that's not good. All right, so closing everything. Now we're gonna see if this guy is gonna turn red too, or pink. All right, so we're going to use the up to 10 minutes, okay? We see that the other one, the negative control, is already turning negative. All right, we'll see. I'll come back in a minute. It's been a couple of minutes, and this one is starting to turn. This one's the Neisseria. It was kind of hard to tell, so I had to get a paper towel to put behind it. So here's the Moraxella on the right. Neisseria is on the left. It's turning red, so that would be a true negative as well. So we have true negative on the negative control. That one's super duper pink. It's got the stick in it, right? Then we have the other two. This is your true positive, which is your Moraxella catarrhalis, okay? And then you have your Neisseria is a negative, true negative, so we'll put those over to the side. Visually, you can see that. And the left one's on the control. Oh, I forgot to start that back up. So there you go. That is how this works out. So again, this would also be done on the GNR section. It's not just the GNCs, but it can be done on the gram-negative bacteria. This is not the nitrate reduction test you will be doing with the anaerobes, the obligate anaerobes, the last section of our course. We have a disc for that. Thank you so much for watching, and I'll catch you next time. Have a great one. Bye.